Good morning, my name is Enrico Scala from the Istituto Dermopatico dell'Immacolata in Rome, Italy, and today I will briefly present the key finding of our recent study published in Allergy. We investigated the molecular basis of wheat and cereal reactivity, focusing on the divergent role of soluble proteins in gluten-derived allergens. Wheat is one of the most widely consumed foods in the world, but its allergenic profile is complex. Clinical reactions might be triggered by two main allergen families: soluble proteins such as lipid transfer proteins and gluten-derived proteins such as omega-5 gliadin. Distinguishing between these sensitization pathways is crucial, since they are linked to different clinical outcomes, including food-dependent, exercise-induced anaphylaxis. We combined bioinformatic analysis of allergen homology across cereals and clinical and serological data from over 17,000 individuals tested by multiplex platforms. This allowed us to explore not only the prevalence of wheat allergen sensitization, but also its structural relationships with other cereal proteins and the associated clinical phenotypes. Our main findings were threefold. First, molecular analysis showed that 3A14, the wheat LTPs, share high sequence identity with homologous in maize and other cereals, supporting a strong potential for cross-reactivity. Second, in epidemiological terms, only about 4% of patients were sensitized to wheat molecules, with 3A14 as the most prevalent allergen. Polysensitization to both soluble and gluten-derived proteins was uncommon, indicating distinct sensitization pathways. Third, clinically, 3A14 and 3A19 were independently independently associated with several reactions and severe reactions, especially with dependent exercise-induced anaphylaxis. In contrast, ZIA-M14 alone was more often linked to moderate symptoms resembling the LTP syndrome phenotypes. In conclusion, our study highlights the dual phase of wheat allergy. Soluble versus gluten-derived allergen follow largely separate pathways and carry different clinical risks. 2F14 emerges as a key molecular marker, not only for wheat reactivity but also for risk stratification of severe anaphylaxis. These findings reinforce the value of multiplex molecular diagnostics in distinguishing true wheat allergy from cross-reactivity and in guiding precision management of cereal allergic patients. Thank you very much from Italy and see you for the next step.